a horizontal section, and if you were to take it pretty much at the level of your eyes and cut through, this is, and looking from the top down, this is what you would see. In the center of this area is the location of the midbrain. And in particular, quite obviously, you can see those two dark bands here. On the right-hand side is a zoom in of this, and you can see the dark band are actually the area where the substantia nigra dopaminergic neurons are located. They are dark, and they give the name of the structure of substantia nigra, so black substance. Because as dopamine neurons age, they start accumulating a black pigment called melanin. Interestingly enough, if you compare the substantia nigra or the midbrain area of a control patient with an age-matched Parkinsonian patient, you can see that the midbrain is actually kind of more whitish, and the two dark bands have disappeared. And the reason is that the dopaminergic neurons that normally are located here are completely gone, and they disintegrate. Lack of cell also means lack of function. And so on the bottom area here, you are going to see two PET scans. On the left-hand side is a normal individual. This PET scan has been taken with a radioactive substance that binds to the terminals of the substantia nigra cells. And so nicely you can see the two dark red bands that correspond to the black bands of the substantia nigra. That shows you strong activity in these cells. However, you compare the left PET scan with the right one, which is taken in a Parkinsonian patient, you can see that those dark, nice red bands have disappeared because the neurons and the terminals are no longer there. And so as the cells disappear, also functionality, we'll see, disappears. What solution can we offer to Parkinsonian patients these days in order to supply to this lack of control of movement? Well, unfortunately, nothing. There is no cure for this disease, like it is often the case for some of the major neurodegenerative disorders. We have, however, some interventions, both pharmacological and surgical intervention, that can help improve the life of these patients. And so pharmacologically, we could give them L-Dopa, which is the precursor of dopamine. And by providing this drug to these individuals, their leftover dopaminergic cells can actually take L-Dopa and transform it into dopamine, and therefore use it as their natural neurotransmitter. An alternative is to give them dopamine agonists, which is drugs that look like dopamine and will therefore act on the postsynaptic terminal the same way and turn on the target of dopaminergic neurons. There are also some drugs that function on the non-motor symptoms, and I decided for now to skip that part, so I'm happy to answer some of the questions later on. But there are other drugs that can be given to patients to pretty much compensate for some of the other symptoms that they have. A more dramatic type of solution is a surgical intervention. And right now we have one possibility. It's called deep brain stimulation, which is pretty much the injection or the implantation of a needle in the nigrostriatal pathway that provides electric stimulus and therefore can replace the functional dopaminergic neurons and supply electricity in order to try to stimulate the function of the postsynaptic cell. Unfortunately, a very, very small group of Parkinsonian patients respond well to this surgical intervention, and there is a lot of other limitation with it, which is sometimes rejection of the implants. But that's one possibility. However, one of the big dreams that everybody has is, if the cell have disappeared, is there a simple way to replace them? And so for a long time, people have dreamed about the possibility of just coming back in and replacing the lost cell. And this kind of possibility has been enhanced by the discovery of the field of stem cells. And so one of the aim, although the latest, maybe it's no longer latest these days, aim of the research on Parkinson's disease is the idea of recreating in vitro in the lab functional neurons capable of becoming mature dopaminergic neurons, and therefore then being able to then replace the cells that the patients have lost. And so the idea is to take all the stem cells and provide them with signals that we are aware of, that help the stem cells go from the undifferentiated cell to potentially precursors, so immature neurons that will look like it, and then push them all the way to differentiate in mature dopaminergic neurons, expressing, and here we'll talk a little bit more about it in a little bit, but markers, genes that we know are pillar of the functionality of dopamine neurons, such as the enzymes that help make dopamine, or the transporter that help recycle dopamine in the terminal. So what would we do with this recreate functional neurons? Well, we would attempt to transplant them and substitute them in the missing neurons in Parkinsonian patient, which in the left, again, a PET scan of someone who has a reduced functionality in the substantia nigra, with the hope that once those cells are implanted, the substantia nigra would go back on the right side to this original full activity with the pristination of entirely the functionality of all the dopaminergic neurons. Well, to get to the point where we actually can transplant those cells, what do we need to know? So for the success of this replacement, we need to know what are all the developmental steps important for specifying and making dopamine neurons. We also need to know what are the factors that are important to keep up the expression of these key genes that make a dopamine neuron, such as the enzyme that synthesize the neurotransmitter, the transporter that recycle it or pack it into vesicles, and all the other little functional steps that help a dopamine remain a dopaminergic neuron. And last but not least, what are the factors that are important to support the cells that have been transplanted? So what are the factors that help the dopaminergic neurons survive once they are in that location? So those are some of the questions that along the way I've been asking myself. And interestingly enough, what I gather as information along my research pathway is that there is a group of transcription factors called the nuclear receptors that seems to be implicated in all the different steps of this process to go from a stem cell to a surviving and mature dopaminergic neuron. And I hope that I will put together some of the data that I'm going to show you now, some of it that has been presented in some of the papers you had a chance to read. But this is kind of my path to try to help the community come up with a potential replacement therapy system. So the first step in making good dopaminergic neuron is to learn all about the development of these neurons. And so one of the ideas is what do we know about the development of the cells in vivo, and how do we bring this information in vitro? And so here it's a series of images that were actually kindly donated by Dr. Emma Anderson. She was a PhD student in the lab in Sweden, in Karolinska, where I used to work for. And so in her thesis, she put together this proper main key images. And so you can see on the left-hand side the midbrain of a developing embryo. And as you can see in the center course where the turn is occurring, at the ventral part of that turn, that's where the midbrain is located. And in that ventral part, so the whole turn is the midbrain, but in the ventral part is where the dopaminergic neurons actually are born. And so the next image is that of an embryo, of a mouse embryo at E12, which is doing kind of the major push for dopaminergic development. And we have taken coronal sections of the tube right in this area. And you can see it in the dark image. This is what the neural tube looks like. And in the white little box at the ventral part of it, that's pretty much the location of dopaminergic neurons. And so what the lab in Sweden, where I used to work in, spent a lot of time doing was to try to map the different subpopulations of cells that are present in this area. And so on the right-hand side, you can see a schematic that represents exactly that area, where we see in the top part of the ventricle. And so you can see three main populations of cells that through development populate and will give rise to the substantia nigra. The first cells are the ventricular cells located in green on the top close to the ventricle. Those are called the progenitor cells. Those are the proliferating cells, the ones that are capable of keep increasing the population of cells in the area, and are also called radioglia cells, who also are capable of self-proliferating, but also giving rise to the new immature neurons. Those radioglia cells are located in the ventricle, but they have an extension that go all the way from one side, the ventricle, toward the pia mater to the outside of the neural tube, creating a scaffold along which the new immature neurons that are born, here located in blue, will migrate. And so they migrate from the ventricle down toward the pia mater as immature neurons. Once they find the right level in this area where they are supposed to position themselves, they will start migrating laterally. And as they migrate laterally, they mature and become finally mature dopaminergic neurons. So what we have been able to do is kind of isolate the three populations and look at gene expression. And you can pretty much barcode each one of these populations, and you can see the panel on the bottom labeled as B, where you can see the three population of cells: the dopaminergic progenitor that are still capable of self-renewing, the immature dopaminergic neurons in yellow, and then those immature neurons will then become mature dopaminergic neuron. In all three cases, they express different genes, and in particular, different set of transcription factors. And that helps us identify the population and trying to understand what is the role of each one of these transcription factors in the functionality of that cellular stage. So far, so good? Okay. So what we have been able to identify is by pretty much mapping the transcription factor expressed in every one of this population, is discover that there are several members of one family that are represented in each one of these populations. And those are the nuclear receptor. It's a very large subfamily of 46 transcription factors. Because they are a family, they tend to have commonalities. They look very similar. They have repetition elements that are common between them. And they tend to function the same way, which is they like to dimerize to find a partner of the same family. And when they form these dimers, then they bind to DNA. They recognize very specific sequence called response elements. They bind to this response elements and influence the way transcription occur on their target genes. Many of these nuclear receptors are actually, if not all of them, are activators. And therefore, once they bind as dimers mostly, they will turn on expression of the genes in which promoter they actually sit in. Many of these dimers are in this family can be either homodimers, and therefore two identical proteins binding to each other and forming an activating complex, or some of them have as a binding partner another member of the family called retinoic acid receptor, RXR. Okay, so most of them work this way. However, there is a very small group of these transcription factors who want to be different, and although they look very much like the partners, the other partners, and they recognize similar response elements, they like to work alone as monomers. The other characteristic of these transcription factors is the fact that they are receptors because they have a ligand that binds to them and turns them on. And so usually the way it works is that the ligand, which is usually a lipid molecule, binds to the receptors, induce a conformational change. This receptor then finds its partner, and once the two partners are bound, they then interact with DNA and turn on gene expression. One of these interesting receptors called LXR, or liver X receptor, is one transcription factor that we mapped in early dopaminergic neurons in the ventral midbrain. And the second one that we knew was expressed and we were quite interested in is a monomeric transcription factor of the family called NER1. So this is the data that I'm going to try to present you, how we worked on two of this transcription factor to show how they actually influence the development of dopaminergic neurons. So what we came to discover is that the liver X receptor, also although called liver receptor, is actually quite important for pushing dopamine progenitor cells, so the ones that are capable of proliferating, into immature neurons, while neuron seems to have a function more in the second part of the development, which is going from immature neurons to mature dopaminergic neurons. So when I joined the lab in Sweden, I started partnering with a PhD candidate who had just done qPCR on the 3 population of cells and had identified this liver X receptor and couldn't make sense of why LXR was there. And so the first thing we decided to do, and I helped doing, is to try to map the expression of LXR in the developing midbrain. And so what we did is we took whole embryos and we used a technique called in situ hybridization, in which you in situ within an entire system, you actually try to detect the mRNA of these genes. And we were able to locate both forms of LXR, LXR alpha and LXR beta, in the ventral part of the developing midbrain. So both LXR alpha and LXR beta are strongly expressed in this area. And here as 10.5, you can see the developmental stage in which we look. So for those of you who are familiar with mice, good, it's a review. For those of you who do not know, the gestation of a mouse is about 21 days. And the peak of induction of dopamine development, so when dopaminergic neurons are going to start popping up and then developing, starts around E10.5, E11, which is halfway through. And so what we notice is that very early on in the decision to become a dopaminergic neuron, LXR alpha and LXR beta were strongly expressed in this area. And then if we cut through the tube and look at it in sections, so here on the bottom part, you can see a schematic of what the neural tube will look like. And the core is the ventricle. And in red, we have located where the dopaminergic neurons would be when they are developed. Okay, and so what we notice is that by looking by in situ again at E11 is that LXR alpha is strongly expressed in the cells that are right at the ventricle area. Therefore, the progenitor cells, the cells that are still capable of proliferating. And instead, LXR beta is much more diffused throughout the tube, suggesting more that it might be present in the immature neurons before they move on to become dopaminergic neurons. And so interestingly enough, what you can see is also that LXR beta seems to be absent from the ventricular cell, where LXR alpha is instead very strongly expressed, suggesting that maybe they work together, but not in the same cells. So then the next thing that people do when you see expression of a gene in a special location in the brain during development, the next question is, what is the function of that protein? And the way to do it is to ablate the gene and therefore create a knockout mouse and look at how the animal developed in the absence of that gene and therefore of that protein. We were lucky enough in Sweden that the lab not very far actually had an animal model of a double knockout of the LXR alpha. And so Jan-Åke Gustafsson was very kind to donate to us early embryos from E10 to E14 of double knockout LXR alpha, LXR beta mice. And so what we did is instead here we look by immunohistochemistry to look at the protein expression of markers for neurons and compare in the top panels a wild type animal and in the bottom panel the what it would look like in a double knockout mouse. And so the first set of pictures on the left show you when we try to look for markers of neurons, early neurons like TUJ in green. And what we saw is that in the wild type animal, in the ventral part of the midbrain, you see this nice organized band in green of TUJ positive cells, therefore immature neurons. Interestingly enough, in the double knockout, there are a lot less TUJ positive cells. When we looked much more in the entire area, what we saw is, in fact, TUJ is normally well spread throughout the ventral area. You can see a lot of red in the next set of panels. And instead, in the double knockout, you can see that there is a much reduced number of TUJ cells, suggesting that there is a lot less neurons present in this area. And when we look at specifically the fate of these immature neurons, how many of them are actually Th positive and therefore dopaminergic neuron positive cells, we see that in the wild type animal on the top, there is this nice thin band of green Th neurons, so dopaminergic neurons. And instead, in the double knockout, there is a lot less green positive cells. And in addition, they are totally disorganized. So somehow we have fewer neurons born and fewer, therefore, of them that become dopaminergic neurons. The next question was, why? Can we tell what is the mechanism? And so what we came to conclude is that actually through a series of immunohistochemistry, which I'm going to describe in a second, we came to find out that the cells, in the absence of LXR alpha and beta, the cells remain immature. So they remain more in the progenitor state. And so what we did is we look at a marker, phosphohistone 3, that normally mark mitotic cells. And in the top panel in H, you can see that there are few mitotic cells in the wild type animal at E13, so a few days after, toward the end of the development of dopaminergic neurons. And instead, if you compare the number of mitotic cells in the double knockout, you can see on the bottom panel that there is a lot more cells. They are still proliferating or having proliferative capacity in the double knockout. And sure enough, when we look at the marker of these neurons at E13.5, so at the end of the neurogenesis period, so the birth of neurons in the midbrain, we use a marker called RC2 that normally labels the radioglia cells, so those progenitor cells. You can see that in a wild type animal, there's very little red at E13. However, in the double knockout, you can see nicely the large amount of progenitor cells still being present, testified by these long filaments that go from the ventricle all the way to the pia mater. In the wild type, there is no progenitor cells at this stage in 13.5 because all of the cells have matured into Th positive neurons. The right panel shows you, in fact, the number of green cells for tyrosine hydroxylase. And you can see that very little red is present, but a lot of mature neurons are present. On the other hand, in the double knockout mouse, there is still all the red filament, and there is a lot less green mature dopaminergic neurons. And therefore, this confirmed that somehow the absence of LXR alpha and beta prevent the cell from making that transitional developmental stage to the more mature precursor cells. It's still left there. We do not know why. We still need to figure out what are the target genes that particularly are regulated by this process. But we know that LXR alpha and beta prevent the absence of those proteins, prevent the turning off of genes that normally push the cells to start to become immature neurons. Well, I went through all this developmental information, but again, if you remember, my original question is, what is the information we can take from development to actually apply to stem cell biology and try to create then dopaminergic neuron in vitro? And so now that we knew that LXR alpha and beta were so important in these cells, we were trying to think, how can we take that information and put it into the in vitro in the dishes to help create in vitro more dopaminergic neurons. One of the good things about nuclear receptors is that they are activated by ligands. Ligands are soluble molecules. They are easy to add to a dish. And so what we tried to do is we took the LXR ligand and see if by using these molecules, we could improve the development of dopaminergic neurons. And so I was lucky at the time to work in Sweden, where there's just about access to any model you want. And so what we were able to do is then take mouse embryonic stem cells and also human embryonic stem cells and provide them this new soluble factor, this new possible signal to see if that would increase the differentiation capacity of the cells and would make more dopaminergic cells in the dish. So I'm going to walk through one by one. In the top panel is the mouse embryonic stem cells. This is immunohistochemistry of independent dishes that have been, in the control case, provided all the normal signal that we use to differentiate a stem cell into a dopaminergic neuron, okay? The immunohistochemistry that was done on them is in red, TUJ, the immature neurons, and in green, again, TH for specifically dopaminergic neuron. And so we did the immunos, and then we counted how many TH positive cells were present out of the entire dish. So in the second set of cells, what we did is we took exactly the same neurons with the same signals, and in addition, we added to them 22-hydroxycholesterol, which is the ligand for LXR. And then what we did is we counted the number of dopamine cells. All the way at the end of the immunohistochemistry, you see a graph, a bar graph, that represent the different conditions, and we counted there the number of TH positive cells out of the dish. And so each column represents how many TH positive cells are present in each one of the dishes. Okay. So the third condition, sorry, let me back up. And so if we look at the columns here in the graph, you can see that in control conditions, normally we get about 10% of dopaminergic neuron out of embryonic stem cells differentiation process. When we added 22-hydroxycholesterol, we doubled the number of dopamine positive cells, which was already a good result. One thing we thought about is, can we force the cells to express the protein LXR beta? And so we took another dish of mouse embryonic stem cells, and in there we overexpressed LXR beta. And when we see by immunohistochemistry, you can see all this very pretty yellow cells, double labeled TH and TUJ. And those, just by overexpressing the receptor, we were able to count again to induce a large increase in number of dopaminergic neurons in these dishes. And last but not least, what if we make the cell express the receptor, and in addition, we give it the ligand? We reached about 35% of TH positive cell in the dish. So what is this data telling you is that we can enhance the number of dopaminergic neuron present during the differentiation process if we push the cell to make more LXR and we activate these proteins in the dish. We obtain very similar results by using human embryonic stem cells. In this case, we only use the ligand, increasing doses of the ligand from 0 in the first panel, 0.1 micromolar or 0.5 micromolar. And actually what we discover is that human differentiation is even more efficient than mouse differentiation is in the presence of the LXR ligand. And when we quantify the number of TH positive cells out of the whole neurons, you can see that we went from about a 20% in the absence of the ligand to about a 60% cells in the presence of LXR ligand. So we triple the amount of dopaminergic neuron present in the dish, which is quite a result. And so, to summarize all this, while I was in the Karolinska lab in Sweden, we were able to place LXR as an important factor in the development of mouse brain. We discovered that in the absence of LXR, we prevented the initiation of dopaminergic neuron neurogenesis, so the birth of dopamine neurons. And we discovered that the ligand for LXR helped promote the differentiation of these neurons from progenitors into dopaminergic neuron. And so that has allowed us to place LXR in the new developmental path of dopaminergic neurons. And so now we actually add LXR to the differentiation process all the time. The world of our screening of all the nuclear receptor present in the dopaminergic neurons also pointed to another factor called NER1. Another lab in Sweden at the same, several years ago actually, had identified NER1 as an important marker for dopamine cells development. And so Zetterström and his colleague, Dr. Perman, in 1997 published a paper in which they placed NER1 as a new transcription factor important for the development of midbrain dopaminergic neuron. So here's a series of images taken from their paper, in which in A, they do immunohistochemistry and, pardon me, technology. They show that NER1 is expressed in the ventral midbrain, that term at E10.5, okay? And what they show also is that when they create a knockout for NER1, they can show that ablation of NER1 completely prevent the expression of tyrosine hydroxylase, and therefore prevent completely the development of dopaminergic neuron. This was pretty much the first important transcription factor that was placed on dopaminergic development pathway for about 20 years. People had not discovered anything so new and so revolutionary in some ways. And a lot of attention was put to the factor Neur1 in the hope that it would be the solution to Parkinson's disease. So I'm just going to quickly summarize here some of the data, but without showing you the role of Neur1 in development, because has been already shown by other people. But so it was shown that Neur1, another factor of the nuclear receptors, was expressed in mouse developing neuron, that the lack of Neur1 expression prevented the development of dopaminergic neuron. And I was able, and I'm not going to show you that data, to show that Neur1 actually inhibits the activity of transcription factors that are important for the proliferation of dopaminergic neurons. So once Neur1 is expressed, it pushes cells from the proliferative state all the way to the neurogenesis path. And so it provides a signal to push cells to become immature neurons. What I was interested is understanding why and how actually Neur1 affect the expression of these neurons. And so here is a schematic of a terminal of dopaminergic neurons of the substantia nigra. I'm pretty sure you guys are all familiar with the genes that are present in the spaces, but just to give you a summary, we know this is a dopaminergic terminal because there is the enzyme tyrosine hydroxylase and the enzyme amino acid decarboxylase, who take tyrosine and transform it into the neurotransmitter dopamine. Once dopamine is made in the terminal, it's actually loaded into vesicles using the vesicular monoamine transporter, VMAT. Okay, so dopamine is loaded into the vesicle, and now the vesicle can go to the plasma membrane and dopamine can be released in the terminal. Once dopamine is released and it does its job at the postsynaptic terminal, dopamine will be recycled as quickly as possible into the terminal through a protein called the dopamine transporter. The DAT, or the dopamine transporter, is what we consider the most specific marker for the terminals of the substantia nigra neurons, okay? If you see DAT present, you know you're looking at a terminal of a dopaminergic neuron in the substantia nigra. This happens to be the protein and the gene in which my PhD lab was actually working on. And so when I joined this lab, we started to work on that. And our idea at the time was Perman and Zetterstrom had just published that Neur1 was important in mouse dopaminergic development. And our question was, if Neur1 is so important in the mouse, is it also important in human brains? And so what we did was trying to localize Neur1 in human midbrain sections. And so here on the left-hand side, you see a section of a human midbrain. In this case, we used in situ hybridization to determine the expression of the dopamine transporter by mRNA. So in situ here, you can see the two nice bands of the substantia nigra, because that's where dopaminergic neurons are located. And then what we did is we did in situ hybridization using Neur1 as a probe to see if Neur1 was present also in adult human neurons, dopaminergic neurons. And here you can see the bands of Neur1 localized exactly in the same area where that is present. So this suggested that Neur1 was a transcription factor that was also important or present in human midbrain sections. What in the lab of Dr. Michael Bannon we had spent a lot of time doing was, what are the conditions or what happens to the expression of the dopamine transporter in humans? And what we know is that all of us, starting at the age of 50, start losing expression of the DAT in dopaminergic neurons. What is also known is, early data from the lab, that in cocaine abusers, the dopamine transporter gets shut down way earlier. And so that you can compare age match cocaine abusers versus control and see that the dopamine transporter is no longer expressed already during the third or fourth decade of life. So what we wanted to know is, as much as there is a correlation between Neur1 and dopaminergic neuron in mouse, is it possible that there is a correlation between Neur1 and dopaminergic neurons in humans? And so the way we did it is by looking at conditions where we know we lose that expression. And so here what we saw is, again, by in situ hybridization, we compare the brain of a control patient and compare it to a cocaine abuser on the top panels, okay? So the dark images are a way of doing in situ hybridization by looking at grains. In this case, on the panel A, you see the grains that reveal the expression of Neur1 in adult human midbrain. On the right side is a cocaine, is a section taken from a cocaine abuser. You can still see dopaminergic cells located, dark in red here, which is the melanin positive cells. And however, you can see that Neur1 is decreased in expression in these cells. So it seems that Neur1 is loses expression, just like the dopamine transporter loses expression in certain conditions. The lack of mRNA is correlated with the lack of Neur1 protein as well. And so we did by immunohistochemistry, we tried to reveal the presence of Neur1 in human brains. And here is a positive dopaminergic neuron. The golden color of the cytoplasm is actually due to the melanin. And in the core, you can see how the nucleus is darkly stained in purple due to the presence of the Neur1 protein in the nucleus. Compare panel C to panel D. This is a dopaminergic neuron taken from a cocaine abuser. And you can see that the melanin cells are still present in gold. And in the middle, the Neur1 expression is decreased in these patients. And so, Neur1 goes down just like dopaminergic markers go down in certain conditions like cocaine abuse. Now the question was, why? And so my job as a PhD project was actually to clone the promoter region of the Neur1 gene and to see how Neur1 is expressed. How does Neur1 eventually regulate the expression of the dopamine transporter promoter? So what I did, I cloned about 8,000 base pairs of the promoter region of the dopamine transporter. And I looked at the elements, the response elements for transcription factors that were present in this promoter. And I was able to locate several elements that normally are recognized by Neur1 in this promoter region and bind. And so what we wanted to know is if those Neur binding response elements, also called NBREs, are functional and therefore are the reason why Neur1 can maintain or not the expression of the DAT. So to do so, there is a kind of a simple molecular biology experiment that can be done. You can take the promoter of a gene and clone it in front of the firefly luciferase gene. The firefly, if you know, at night emits light. And so you can take that protein, the part of the coding gene, and put it next to the promoter of interest of yours. And so imagine that here is the promoter region. When transcription factors bind to that promoter region and open up the chromatin, then you turn on transcription, and therefore you turn on transcription of the luciferase gene. When you provide the right substrate, if this gene is functional and the protein exists, it's going to start emitting light. And so it's a simple assay to look at if a transcription factor binds to the promoter and turns on transcription by just measuring how much light you can actually see in the assay. And so what we did is try to ask the question, is Neur1 important for the expression of the human dopamine transporter gene? And so what we did is we took the large promoter region that we had identified, and here you can see the location of the potential NBRE sites, the little white boxes in the promoter region. We cloned that promoter in front of the firefly luciferase, and then we transfected cells in the presence or absence of different amount of Neur1. And here is a graph that shows you on the x-axis is different quantity of Neur1. In the y-axis is the amount of light that is emitted. And what we saw is that by comparing an artificial promoter, where we only had the perfect Neur1 binding site, 3 of them connected to luciferase, and we compare therefore, the light emission of this artificial promoter, compared it to the human dopamine transporter promoter in the presence of different amount of neuron. And as you can see, as we increase the presence of the transcription factor, both promoters are strongly activated and started to emit more and more light. They do so in a very similar manner, suggesting that the way neuron binds to the NBRE is very similar to the way it binds to the human dopamine transporter. And so neuron is actually an activator of that expression. And we were the first one to be able to demonstrate that neuron is important for the maintenance of the key genes present in dopamine terminals, like the dopamine transporter. So neuron is expressed also in adult human brains. Neuron is expressed in similar patterns to important markers of dopamine neurons, like the dopamine transporter. And neuron positively regulate the expression of some of these key genes. What I was left out when I finished my PhD was, yes, okay, neuron turns on that, but how does it do that? And therefore, I decided to continue my research career trying to understand how does neuron interact with the DNA of the dopamine transporter, and how, therefore, does it induce transcriptional activation. And so what I did, I went to a lab in France. It was a lab that was a biochemistry lab where they studied how transcription factor bind to DNA and what are the factors around it that are important to support transcriptional regulation. And so what I asked there is, what controls the binding of neuron to DNA, and what are the factors that are important to interact with DNA in order to open up chromatin and allow the expression of the genes. And so when I went to France, I changed completely the type of research I was doing, because for the first time I was exposed to the world of protein work, protein research. And so my idea was to try to find other proteins that interact with neuron and therefore enhance transcription. And the way I did it is I looked at different types of cells and tried to find some cells that I could grow in a dish that also expressed neuron. And so here you see a RT-PCR in which cellular extracts of CSM cells derived from the midbrain express neuron. You can see the white bands here. And they express neuron just like the midbrain, in this case of a rat, would do. Okay, so these cells make neuron. Neuron in this cell is transcriptionally active. And so what we did is we took these cells and we transfected them with the artificial neuron binding sites, and we added increasing amount of neuron. And we saw that neuron was capable of inducing activation of genes. Here you see a bar graph that shows you that you see fold increase in activity as you increase number of neuron, a quantity of neuron. So now I had a model that expressed the factor, the transcription factor I was interested in, and also that in a model in which this transcription factor was transcriptionally active, was capable of regulating gene expression. And therefore, within those cells, there must have been all the other proteins that were important to help neuron function. And so what I tried to do is to identify what are those proteins that bind to neuron and help him work. So the way I did it, I created, and this is the right part of the slide, a bait. I created a construct that had neuron as a protein in its normal conformation, and I attached it to some beads, locked them up. And then I took the CSM cells, I purified them, and I took the nuclear extracts from these cells, so that all the proteins that are present in the nucleus of these cells were pure and simple. So then I incubated together the neuron and this cellular extract to try to see what were the proteins that remained stuck to the neuron and therefore could be important factors to helping function. Once I incubated those, I then released the nuclear extract that were attached only to neuron. I washed off everything that was non-specific, and whatever remained attached was separated in a protein gel. And here you can see a gel. So in this gel, we started to separate protein by size to try to see everything that could be an interactor of neuron. And then I worked with a lab that was an expert on mass spectroscopy in order to identify what exactly each one of these bands present in a gel corresponded to try to identify what proteins were present. And this is what we identified. A lot of different protein, but they were mostly category that was very interesting to me. So we identified transcription factors that could be partners of Neur1. If you remember when I told you initially about Neur1, I told you that is a member of a family that normally works in dimers. But Neur1 seems to be special. He seems to work on his own as a monomer. It turns out that it can also work as a dimer. And here are some of the other transcription factors that can interact with Neur1. And several of them are transcription factor of the same family, the nuclear receptors. We also identify transcription factor that repress transcription. We identify potential DNA repair molecules that could be important to help in fix DNA and therefore help cells that are in danger of cell death to actually be repaired. But importantly, we identify a large group of signaling molecules. Why is this important, or why this seems a possible breakthrough? Because Neur1 is part of this family. This family normally has ligands that bind to this receptor and turn them on. Neur1 not only works as a monomer, but so far it's orphan of ligand, and therefore we have not identified what is the mechanism that turn it on and turn it off. There has been no molecule identified to do so. And so one other way of turning protein on is to add phosphate groups to them. And so one possibility is that signaling pathway turn on Neur1 by adding a phosphate group to it, and therefore change the conformation of the protein and make it turn on. And this would be an important breakthrough to help us activate Neur1 within the dishes in the differentiation process. And so in particular, what I focused on is one signaling pathway called the ERK family. And I'll move on quickly to tell you why I focus on this. Because one of the members of this family, ERK5, is important for survival. And so this brings back to the three questions I was interested at the beginning, which is, what are the developmental knowledge that we need to have to make better dopaminergic neurons? What are the factors that are important to express dopaminergic neurons? And at the end, what are going to be the important factors that we need to provide to the cells to survive the transplantation study? And so the idea of identifying an important survival factor like ERK5 makes us think that maybe Neur1 can be used to turn on gene expression of dopaminergic neurons and make them survive in the dish. And so ERK5 has been highly expressed in the brain. It's implicated in neuronal development. And it's possibly expressed in the midbrain area. And here on the bottom, you can see a comparison of wild-type animal and an ERK5 knockout animal. And you can see that the neural tube in the midbrain area that turn is strongly affected in the absence of ERK5. And so what we wanted to know is it possible that ERK5 and Neur1 work together as proteins in order to turn on genes in dopaminergic neurons? And so in the French lab, I looked at how these two proteins interact, and I use a technique called immunoprecipitation, where just in simple fact, it's a Western blot. But what you can see is that when Neur1 is present and ERK5 is present, the two proteins are in the same transcriptional complex. When the two proteins interact with each other, we can, technical issue. When you add ERK5 to Neur1, you can see that Neur1 is increase in transcriptional activity. And I'm sorry, this graph has been cut out, but I'll show you a different one. So I'm going to walk through the last graph just to make you understand. So I took cells, I transfected them with Neur1 and the luciferase gene. And what you see is that in the presence of Neur1, you increase the activity of the gene. When you add to Neur1 different doses of ERK5, you can see that Neur1 becomes more efficient at inducing light emission. This functions only when ERK5 is a functional kinase. And you can take a constitutively active ERK5 and add it, and you can see that neuron activity is enhanced when you use constitutively active ERK5. However, when you take a kinase and you mutate the kinase domain, and therefore the ERK5 is no longer capable of adding phosphate group to neuron, you can see that neuron goes back to this normal transcriptional activity. This data suggests that not only the protein interact with each other, but ERK5 is an enhancer of neuron as transcription factor. And so this data, although in vitro, it helps explain or possibly place neuron and ERK5 on the survival cascade for dopaminergic neurons. Now, I'm almost done, and I'm just going to show you what we have been doing at UHeart on this interaction of ERK5 and neuron. So all the data that I've shown you until now is been found in vitro, in biochemistry, in cell lines. So what I have started to ask here with my both master students and undergraduate student is ERK5 important for dopamine development? And so can we go back into the developmental process and look if actually ERK5 and neuron interact together in the brain of mice? And so here is a regular PCR with the different developmental stages, E12, E14, and P0, which is when the mouse are born. And we look for expression of dopaminergic markers, tyrosine hydroxylase, neuron, and ERK5. And you can see that both during E12, E14, and P0, in area where TH and neuron are present, therefore in dopaminergic neuron, you can also see expression of ERK5, suggesting that ERK5 is present in the mouse in the right place at the right time. And here is the latest data we just got before we got shut down, is that by qPCR, we looked at the developmental profile, the expression levels of neuron and ERK5 during the entire development of dopaminergic neurons, starting at E11, the beginning, until we get to adulthood. And what you can see is that neuron is highly expressed in E11 in E12 and E14, and then as we come adulthood, neuron expression goes down. Differently though, on the right panel, you can see that ERK5 is present in E11, E12, and E14, however, starts to strongly be increased in adult. So right now I cannot tell why, but all I can say is neuron and ERK5 are expressed also in vivo in the brain. They seem to be potentially expressed during the developmental process as well as during the maturation and the maintenance of adult dopaminergic neurons. So in summary, what hopefully I show you is that you can go from gene expression data in vitro to stem cell biology. I hopefully show you that nuclear receptors are a large family that have an important role in the development of dopamine neurons. They control the development of dopamine neurons. They regulate the proliferation of the progenitor cells through LXR. They control the functionality of dopaminergic neurons. They help in the maintenance of key genes, TH and DAT. And also they seem to control or being correlated with human disease conditions. And also it seems to be possible that nuclear receptors are important for the survival of dopaminergic neurons and could cooperate with survival factors like ERK5 to maintain the expression of these genes. And with this, I hope that we can use all this information to actually make better dopaminergic neurons in a dish to be able one day to reach the idea of transplantation studies to improve the life of Parkinsonian patients. And I'm going to skip the last little part. Sorry, we can go back to this. This is what we're doing now. But I would like to acknowledge the people who have allowed me to do some of this work. The data that you have seen, the dopamine transporter data, comes from my PhD studies with Dr. Michael Bannon in Detroit. The biochemistry of the ERK5 pathway comes from Dr. Philippe Lefebvre in France. The data on the LXR comes from Dr. Ernest Arena in Karolinska Institute in Sweden. Along the way, I was lucky enough to get some funding. And all this work has been done because I cooperated with a lot of undergraduate students and graduate students, both at Mount Holyoke, where I worked as a visiting neuroscience faculty member, and also at UHeart, with the help of graduate students and undergraduate students in both the biology and the neuroscience department. And so with this, I thought that I'd give you enough information to maybe ask any kind of questions you want to know, both about my career path as well as my research path.
Thank you, Dr. Sacchetti. That was great. Students, if you have questions, feel free to unmute yourselves and just ask her. If there's a reason you can't because of background noises, it's also okay to type in the chat box, but it's better if you can unmute yourself just because then we can interact a little bit. But first, just thank you so much. That was really...
You're very welcome. I hope that it was a lot of molecular biology, but I hope it's kind of okay.
Oh, but that's good. That's good.
Hi. Thank you for coming to speak to us. We all appreciate it. I missed the beginning of this talk, so if you might have already discussed this. But I was looking at your staff page on the UHeart website, and you were talking about designing educational tools to inform the community about these mild traumatic brain injuries.
Right, so this is, yes.
So what have some of these tools that you've been developing, and what about maybe the development in the future? And then specifically with these brain injuries, when these for example, you study dopamine pathways, and how does this affect potentially the development of these pathways?
Right, so the two topics are a little different. So in the website, there is a description of my dopamine or my research in neurological disorders. And then a kind of a newer interest of mine that I have is the idea of trying to better understand traumatic injury and trying to see what we can do. So the talk was not really related to that, but I'll address you what are the tools. So part of it, I'm starting to be very interested in TBI, mostly because in my introductory biology courses and some of the other courses, I have a lot of athletes. And along my 6 years of teaching, I've had a lot of students, athletes, who had to take leave of absences or had to take periods of inactivity because they had concussions due to games or so forth. And so what I discovered is that there is a return to play practice, but there is no return to class practice. And so the athletes would take away about a week or maybe 10 days to try to recover. And then as they came back to class, the biology faculty members asked them to take the 3 exams that they missed, the 12 quizzes, to write the essays. The chemistry department did exactly the same thing, and the students were incapable of keeping up with the work. Sure, Anna, I'll get to you after. And so what I started to do with some of the students that were in my lab who actually were athletes too, start to have conversations. How do you return to play safely, but how do you return to play, how do you return to class safely? And we seem to have discovered that there is a disconnect. And so me as a neuroscientist is very interested in the well-being of my students, and as well, of course, at the well-being of their brain development. And so we started to kind of create, we are at the basic steps, we are creating a questionnaire that we sent to all the athletes and to all the trainers and the coaches to try to figure out, to allow the two parts of this world to talk to each other and determine what is the best way to do it. And so we have therefore discovered that there is a large disconnect, and that it's not so easy to help a student come back to the field. And so with the students, we started to put together some kind of PowerPoint slides to walk through basic information about the brain, that they would present to their coaches at the beginning of the season and help them understand what is known about brain impacts that might actually be affecting their practice. So that's a kind of a separate world a little bit. On the other hand, one of the things we had discovered, we were trying to do TBI in a mouse model of TBI, and it turns out that it's a little bit more complicated than what I thought, because the brain is a mass. And so all the models that people have to do with how do you impact something and see how it changes, but without taking into account that there are 12 structures on the way from the impact to, for example, the substantia nigra. And therefore, we had no real way of understanding how the substantia nigra was directly impacted by the blow to the head that we were doing. And so for now, the actually molecular mechanism of it have been put a little bit on the side. I've gone back to my old world of studying in basic ways, molecular biology. So, but hopefully there are things slowly with time that we can get there. But I think it's an important point, and I think that all us as educators and neuroscientists should figure out a way of making sure that all the components in the university are all talking the same language about return to play and return to classroom work.
Yeah, thank you.
You should hear about Anna's senior honors thesis. Do you want to tell her what you've been doing?
Yeah, so I did the clapping emoji because my thesis is on Trinity's return to play protocol. And so we did the same thing. We surveyed faculty, coaches, and students. And we're analyzing data now, but so far we found that the majority of people in each 3 category didn't know that we had a return to play protocol at Trinity.
Return to learn, not return to play.
Sorry, return to learn protocol at Trinity. And a lot of students felt that they didn't have good communication between their coaches and faculty because everyone was sort of getting different information. And then we're also looking at if specific cognitive deficits correlate with a harder return to the classroom. So that's what we're figuring out now, but it was interesting to hear you talk about.
Right. I think we're all in the same position.
She surveyed, faculty, students, both athletes and non-athletes, and then and coaches and trainers. And if they had a concussion, she gave them some cognitive measures of attention and memory and executive functions, so we can see whether they're predictors of who has more difficulty with return to learn than not. But we actually do have a kind of protocol here at Trinity, and so I would love to talk to you more and see if we can collaborate a little bit and look at how different institutions, because it obviously might work differently at different places, but I think we have the same issues here. We have faculty who are frustrated. Students are overwhelmed. Faculty are frustrated because they're like, I can't create a whole nother class just for this person that had a concussion. 13 weeks is quick. So in high school, return to learn protocols are take 3 months, and get better. And you can't do that in a college semester. So I think you're really onto something. I think it's really important.
Right.
Most of our student responses said that they only returned to the classroom because they were afraid of falling behind, not because they felt ready to do so.
But then I have observed the eyes of the students who were 4.0 for the first 3 weeks or 3 months, and all of a sudden they sit there and they're not there, and they don't know they're not there. And that's terrible. And I think it's detrimental to their brain recovery, and we're forcing them. And as a neuroscientist, I find that absolutely unbelievable. So we need to do better. So keep working.
So you have a chat question, in the chat box. Do you want me to read it to you so you don't have to look it up?
Sure.
So Natalie says, I apologize, I'm not able to unmute myself. What changes would you expect to see in the brain of a person who has only used cocaine infrequently? Would you expect to see a recovery in people who stop using cocaine?
Yeah, so this is one of the interesting things. I actually started to do neuroscience because I was very interested in drug abuse, and then ended up taking a completely different turn, which is okay. But it gives me enough tools to be able to talk about different things, which is pretty good. So one of the things that in Dr. Bannon's lab we were looking at is frequency in some ways. And the idea is we have no idea each one of these individuals how often they use cocaine, but we were able to get their brain when they died of an overdose. So like most of the drugs, you don't die because you're taking cocaine, you're dying because your body cannot handle whatever status or environmental changes they are. And so, for example, the amount of cocaine you were using yesterday when it was 20 degrees below in Detroit gave you a certain effect, and now all of a sudden the next day is 80 degrees and your heart is this size and your veins are dilated. You take the same amount of cocaine and you end up having a heart attack. So we could not do the change, kind of go back and say, how often have you used cocaine and so forth. But what it was clear is that you can categorize the brain of these users, and repetition help the system adapt. And so the system is very plastic, and the more you take cocaine, cocaine actually binds to the dopamine transporter, prevents a reuptake of dopamine, and therefore allows dopamine to be at a synaptic cleft for much longer. Well, the system says, if you keep inhibiting it, why am I wasting all this energy to make this protein out? I'm just going to take it away. By not having it at a terminal, whenever you release dopamine, you're going to have as much dopamine as you want in the terminal, and it's going to do its function. And I, as a cell, avoid wasting a bunch of energy. And so this is what we have seen, is that people that die of an overdose probably have abused cocaine for a long time, and it was clear that their dopamine transporter was not expressed anymore. We also know that in ages, starting at the age of 50, normal humans start to lose expression of the DAT. But that is, again, at longitudinal studies. So I think that the brain system is very plastic, and so if you use a few times, it doesn't actually affect it in the long term. But it's the repetition and the repetition of it is going to have a dramatic effect. So I think that probably using it a few times, the system has ways of clearing and responding to it, just like any other protein. And so maybe for a few months or something, you would maybe see decrease in expression of the protein. But as that demand goes away, then the system recompensate for it. So I don't think that that would be dramatic in the long term. Does that hopefully help Natalie? And then
so Maggie and Roxana both have questions.
Okay, sure, Maggie.
I was wondering, for kind of a clinical application of your work, whether or not there's been work to kind of find a drug that mimics Nurr1, if that's the reason why there isn't a large activation of like this HDAT expression, as well as like activity of progenitor cells, and whether or not there's issues with kind of, I guess, sort of like specification of like what genes like that, like mimicking drug activates.
So that has been, people that know me, I'm the joke, because Nurr1 was what I call the millennial protein. Because when I went to my postdoc, it was the turn, and therefore I was going to discover everything about Nurr1. Turns out that there have been a few studies that have shown that there are some molecules that seem to kind of activate it, but nothing efficiently. And there was another study that did crystallography that suggests to show that Nurr1, the binding part of the protein that normally in other receptors is open so that the ligand can come in and change. It seems that that conformation is always closed, suggesting that there is no pocket for a ligand, really. And that's what made me believe that phosphorylation might be actually, right, the mechanism that turns it on and turns it off. And so around the end of the 1990s and the beginning of the year 2000, all the pharmaceutical companies were all doing Nurr1 studies. I was contacted, and then I worked actually for Sanofi, doing some of this luciferase assay for them to test molecules after molecules, and we couldn't find very much. And so pharmaceutical companies work this way, where if within a certain amount of time you can't find a solution, that's a waste of money, drop the project. And that's pretty much how the field of Nurr1 has stopped. So for about 10 years, everybody thought that Nurr1 was the solution, and because we don't have answers, it got dropped. This is good and bad. Good for me, because then at UHARD, which is a much more smaller place, I can continue to do my study and hopefully bring up something and discover some things. But at the same time, without a molecule is an easy thing for drug companies to work with. And There is right now no answer to it. And probably I would assume that the answer will come from different models. So Neur1 recently has been shown to be very strongly expressed in tumors. And so my assumption is that models where it's easier to grow them in a dish are going to provide some of this molecular mechanism answers, and then we can take that information back to the brain and apply it there.
Thank you.
Sure.
Roxana?
Hi, my question is, how far away do you think we are from incorporating this data that you've discussed on LXR receptors and their ligands on diseases like Parkinson's?
So in the in vitro differentiation, this data actually, at least for the LXR part, is being incorporated as we speak. So at least in Karolinska, which is a lab that is one of the major leading lab in trying to do Parkinson transplantation models, they're using the LXR solubles for that. The idea of the transplantation study for now kind of got put on hold. And the reason is for many years, the Michael J. Fox Foundation, together in particular with a large group of Swedish scientists who were the pioneers in the transplantation studies, have done a lot of work and collected a lot of money and invested a lot of money in embryonic stem cell differentiation into dopamine neurons for transplantation studies. Unfortunately, the results of those experiments were far from being ideal. And a publication, particularly in the 2008, I believe, I think it's a Nature Neuroscience paper from Lund, Sweden, showed that the people who had been transplanted, although they were fetal tissue and not embryonic stem cells, but the people who had been transplanted with fetal tissue, the implants survived, but they started to have accumulation of alpha-synuclein, which is the agglomerate that potentially cause the degeneration of the neurons, suggesting that healthy cells with age get affected by the disease. And therefore, it's not only about replacing the cells. We can keep replacing the cells, but the cells will end up keep being sick.
Okay.
And so we need to discover other things. They are going to be able to help us with that study. And then the other part, the surgeon felt that the transplantation studies were extremely complicated and often expensive and did not give exactly the results that they were hoping for. And instead, that deep brain stimulation was a little bit more rewarding, both for the surgeon as well as for the patient. And given that the results were better with deep brain stimulation, so far they were the ones who said, we are going to halt this transplantation studies and rather push deep brain stimulation and wait until the transplantation studies can give better results. And so that's where it's been kind of stuck for a little bit.
Okay, thank you.
Yeah.
All right. Anyone else?
Yeah, I have a question.
So we have a chat question from Emma, who says, why are there differences in LXR beta and LXR alpha expression in neurons? Is there a difference between, I think there's a word missing, but something different between what they do?
So one of the things that this is exactly where the studies in Karolinska were focused on is LXR alpha and LXR beta look very similar to each other, but there are some obviously protein differences. But they bind to DNA, seems to be in a similar way, and the conformation of the C-terminal is, of the N-terminal is pretty similar. What the in situ hybridization suggested is that alpha is expressed in a certain population of cells versus beta in a different population of cells. And therefore, they probably have different targets. And that difference in activation of a pool of genes versus the other makes obviously the whole difference. There has been no studies done exactly in the mechanism, and there have been no target genes identified. But what we know is that LXR works in the ventricular cells, so the real radioglia cells, and instead LXR beta seems to have as targets the cells that are immature neurons that are starting to migrate down. My hope was that LXR and Neur1 were interacting with each other, and therefore that I could have kind of put the two worlds together and find target genes. We were never able to, and in addition, there is no protein-protein interaction between the two. So there is still a lot of biochemistry that needs to be identified. But clearly, the pool of cells in which they are expressed is different, and therefore they are going to do different jobs. I hope that partially helps.
Okay.
My question is.
It's hard to hear you, Olivia.
Oh, sorry.
Mm-hmm.
Is that better?
Yeah.
Okay. So because there are like other neurodegenerative diseases that involve dopamine, like MS or I can't think of another one, but what made you decide to specifically focus on Parkinson's disease?
Well, because for many years, and this is where the knowledge that we have of Parkinson's disease now is different, but for many years, and that would give away how old I am, in the 90s, everybody believed that Parkinson's disease, it's only a disorder of movement, and that there were no real cognitive issues in these patients. And because it's a disease of movement, the substantia nigra had to be the one that was at fault. And so everybody was focusing on that disease and that pathway, not only because we know where the cells are located and we know the target. It's kind of an easy thing. You have one cell that you need to replace in order to be able to then fix the problem. And so everybody focused on Parkinson's disease because it seemed to be an easy fix. And it turned out that it's really not easy. And one of the reasons is true that we know where it starts and where it ends, but the axons of those neurons is extremely, extremely long. And it's very difficult when you, for example, do transplantation studies to have the axon grow exactly in the right direction. Also because we don't know what are the signals important for axon migration. And so that adds a large complicated issue. One of the things also has to do with the labs where you end up are the ones that form you mentally. And therefore, I was in a lab that was doing drug abuse, and they were focusing on the substantia nigra cells, and it became kind of my home. I was comfortable with those cells. And so there's multiple questions that get added as you study things, but they're always focused kind of on the same thing. And so I grew up and I was groomed to think that the substantia nigra cells are the most important cell in the brain. And I happen to like that idea, and therefore I continue that way. But it's true that dopamine is involved in other things, but the two main things are actually drug abuse and motor control. So that's kind of where I got fixed. At some point when I tried to do my second postdoc, I had an interview with a lab in the New England area at the time. And they were studying retinal development, which is quite interesting because all the transcription factors that are important for dopamine development are actually important for retinal development. But this scientist was very nice to me and said, I want you to understand that if you move into the retinal world, you're going to abandon the world of dopamine neurons. You'll never go back to it. And I had to make that decision. And therefore I decided that my second postdoc was going to be in a dopamine lab rather than completely changing field, because after all, I was very interested in that. So.
Thank you.
Yep.
Anyone else?
Hi, yeah, I have a question.
Mm-hmm.
So I know this is kind of like your future directions for your work, but how would you intend to implement your in vitro work to take steps towards being implemented clinically, kind of specifically with your research on adding the LXR beta and seeing the improvement in the number of dopamine neurons?
So I'll tell you that from the beginning, I'm a molecular biologist, and honestly, what I'm really interested in is understanding the mechanism. And so as everything, I think that we have potential tools, but we need to understand how they work in order to be able to say, oh, we're going to use LXR ligand for Parkinson. We're going to use LXR alpha specific ligand maybe for MS, like the other student was mentioning. And so what I would like to do is be able to first make sure that all the components are really important, expressed in development. Then bring them back to a dish and see that when I add it, you can actually really see the improvement of the number of cells, and that is consistent. One of the problems with dopaminergic neuron differentiation If you recall the graph I showed you, what we know about mouse and human development with the protocols we have now is about 10% or maximum 20% of the cells become dopaminergic neurons. That means that the rest of the dish that we have in our hands is something completely unrelated. And therefore, we cannot take that dish and transplant it into a human brain, because we are going to give them dopamine neurons, we are going to give them unspecified neurons, we are going to give them cells that are still capable of proliferating. And so what it's suggesting is that our protocol, differentiation protocol, is not 100% efficient yet. That means we are missing signals that are important to get to that point. And so I think that there will have to be a lot more bench studies to identify exactly what the pathway is and in which order things need to be added in order to push that. So my goal is actually to be able to, who knows when, but take a dish, take all the signals that we have identified, put them in, and go from 100 cells to 100 dopaminergic neuron. And at that point, I think my job is done. And I will let everybody else do the transplantation part, which is, I'm interested in the mechanism, but the mechanism need to, tools have to have an understanding behind it in order to be appropriate for the job that they have to do.
All right, so it's about 1:45, maybe one more question? Or we're good? Is everyone good?
I could ask a question.
Okay, Russ.
Yeah. Have you ever considered studying the effects of cocaine abuse in the midbrain of offspring in rodent models?
My answer is no, and I tell you why. Because actually, before I got into this molecular biology labs, I worked in a behavioral neuroscience lab, and I did, in that case, morphine. And I was trying to reverse the effect of morphine by injecting protein kinase inhibitors. My experience in the lab after a few months was that every time I came home, I would be in tears for the things that I had done to the animals and for the things I had seen the animals do in response to what I did to them. And therefore, drug abuse and animal models are not my cup of tea. And so along the way, I had to make decisions of what I was okay with. And this is partially also why I steered a little bit more toward Parkinson rather than drug abuse, because all the tools that at the time were available were very animal related, and I couldn't handle it.
Gotcha.
It's not for me.
All right, thank you.
Yep.
Anyone else? All right, well, normally we would take some time to get to know, for you to get to know all of them, but I think under the circumstances, we should probably just say thank you.
You're very welcome. It's been a pleasure. I hope it was not too intense or
It was
overwhelming.
perfect. It was
Good luck, everybody. I know it's a different way of ending, but good luck. And who knows, maybe this summer we'll have a big party and get to know each other.
Yes, perfect. We'll do something together at the Science Center again or something.
Yes. Oh, that would be lovely, actually.
Yeah. All right,
All right.
take care.
Bye, everybody. Bye, Sarah.
Bye.
Thank you.
Bye,
Thank
thanks.
you.
Bye.
Thank you.
Thank you so much.
You're very welcome. Good luck.
Thank you.